So, at this point we'll begin our panel discussion. First I'd like to point out that the suggestions that will be made for the panel discussion are just that, merely suggestions. I'd like the audience to feel perfectly free to participate to whatever extent they would like to participate in this panel discussion, and to discuss anything that they feel is relevant or important. Just a little reminder that the concept of personalized medicine is new, but very often what is old becomes new again. This is a quote from William Osler. He was instrumental in modern medicine in bringing medical students from the classroom into the clinic, and for creating the first medical residency, hospital-based residency at Johns Hopkins University. And again, it's as true today as it was when he stated this. That variability is the law of life, and as no two faces are the same, so no two bodies are alike, and no two individuals react alike and behave alike under the abnormal conditions which we know as disease. So very inspiring to know that we are re-exploring something that has been known for a while. I've broken down a whole series of questions that were submitted by the speakers today through a series of emails, and it rambled on for about 10 pages or so, and we've condensed it down to a whole series of questions. Basically, in 4 categories. 1 being biomarkers, 2, a variation on the first to no harm concept, avoiding the unintended consequences of genomics. Question 3, BRCA mutations. And question 4, multi-parameter systematic strategy for PPPM in cancer. I think we'll start with the biomarkers, and as you can see, we've had several different speakers send in suggestions. This is a compilation of that. Why are there so many published biomarkers yet very few in clinical practice? What is wrong in the validation process, if anything? 2, what are the key challenges with the identification and validation of biomarkers? Which biomarkers are useful? How much do biomarkers account for inter-individual differences in drug metabolism and response? Should molecular profiling, genomics, take precedence over proteomic analysis in initial fishing expeditions for biomarkers? And finally, is pre-surgical genomic analysis of potential value in determining individual patient drug response? So at this point, I'd like to open the session to the floor. Yes?
That's a good question. It's an important question. And I think that as I read the literature and try to do this research, I always come back to, have we identified, if we're talking about a protein, potential biomarker, for example, and I always worry.
And I always worry, have we really identified the protein? What does that mean, biomarker? What does that mean, protein? In a rigorous sense, a protein is only defined by the amino acid sequence. And I don't know of any paper that has got the amino acid sequence of the entire protein that seems to be important for a biomarker. There's a lot of receptor assay, immunoassay, etc., that indicate that they might be specific. But in a rigorous sense, you don't get amino acid sequence from an immunoassay. So therefore, anything that is defined as a biomarker based upon that analytical method, I question dramatically. Now, the only way to get amino acid sequence might be mass spectrometry or tandem mass spectrometry. The sensitivity is dramatically improving rapidly, but not down to the 1 copy per cell level. And there's the gap. So that's the first reason, I don't know which one you have, I think 1A. Can we question biomarkers? Yes. Because we're investing a lot of time, money, big pharma, et cetera, to say it's a biomarker. So one of the reasons why there's few in clinical practices, I don't think we have ever defined a biomarker. And I don't know if there's anything wrong with the validation process because we don't have 1AA done yet. And what does validation mean? Now, how are you going to validate that? By amino acid, receptor acid? I think if you have amino acid sequence, you don't have to validate. It is validated. So I think that's 1A, 1B, and I think they're the very important parts. Thank you.
Anyone else?
I think I disagree. You're talking about the technical validation. And I think the main problem why so many biomarker candidates have been identified and we have not yet validated them is a problem of biology. Look at the diagnostic protein assays. None of them is really doubt or question whether the captured analytes have a variation in amino acid sequence. So that's I think it's really a technical issue. And if you look at very, very carefully at all the biomarker discovery publications, and you can just look at more or less first the Cyphergen list of manuscripts, more than 200 I think. And of course, those need an amino acid sequence to get identified. But whenever you start to develop a sandwich immunoassays, the two specific capture molecules are combined to increase specificity tremendously. You can be pretty convinced that from a technical aspect, such an assay is robust and reliable.